There we go. Now it's working. Okay.
So, do you have the
Key?
key pulled up?
Yep.
Awesome.
Right here.
Alright, so let's just go through them. And
Yeah. I'll start with number 1, I guess.
yeah, I don't have the questions in front of me, so you can answer them.
So, number 1, what are the 3 chemical components of nucleotide? There's phosphate group, the deoxyribose or ribose sugar, the pentose sugar,
Mm-hmm.
and the nitrogenous base, which is adenine, guanine, thymine, uracil, or cytosine.
Mm-hmm.
And then the one prime carbon that's attached to a hydroxide group, and the nitrogenous base in the ring oxygen. That's basically this. 2 prime, I said in DNA it's attached to 2 hydrogens, and RNA it's attached to a hydroxyl group, and 1 hydrogen. Did you see here? So hydroxyl group.
Yeah.
Yeah.
And then that's what distinguishes the 2. Yeah.
Yep.
3 prime, I said attached to a hydroxyl group, helps form the backbone opposite to the 5 prime carbon through a phosphodiester bond. It's the positive charged side of it, and it's important in synthesis.
Yep. That's what.
4 prime, I said it's attached to the 5 prime, and then a methyl group and the ring oxygen. It's just there. And 5 prime, I said it's attached to the phosphate group. It's the negative charge in the phosphodiester bond. And it helps form the backbone with the 3 prime carbon through the phosphodiester bond. And is also important in synthesis, DNA replication and stuff. Yeah.
Yep.
Okay, who wants number 2?
Okay, so this question concerns the structure of DNA and chromosomes. What are the 4 nucleotides that can be used for RNA synthesis? Please write the entire name for each chemical. I put guanosine triphosphate, adenosine triphosphate, uridine triphosphate, and cytidine triphosphate. Is that right?
Yeah, that's right.
That's what
Okay.
you put. And then now that you can also switch it out for
Okay.
di or mono, right? Depending on the.
Yeah, this is just asking for RNA synthesis, so isn't it always a triphosphate?
I don't think so, because
for RNA?
in the PowerPoint it was showing.
I guess
mono.
for like bacteria and things like that, they might have mono, but I think in humans it's always triphosphate, isn't it?
I'm not sure. I don't remember.
What did the key say? I think the key
The
just
key
said
just said
tri.
triphosphate, is all it said.
Mhm.
Here's PowerPoint just real quick here. We have to be 30 minutes anyway. Some minutes long.
Might as well.
Here, some of my pictures I blew up super big so I could actually see them. So I can't. I'm using the instructor notes. Did I say that?
Well, in the instructor notes, it does talk about how it can be like mono or
Mono or di.
di.
So do you know
Right.
do you know for sure if it is in
Humans.
eukaryotic cells? Is it?
I've just never heard of it not being triphosphate.
Yeah.
I mean, I guess it could be something went wrong, and then it's a di. or
Just real quick,
something
I'm
like
gonna
that.
ask Google because Google knows everything. Is it? So I know that I think most of the time it's not see, but it can be like
For like different things,
Mm.
maybe.
DNA, no, RNA.
That's the one I need help on.
What?
This is just my math.
I didn't know how to do. Is that number 3, 4?
4. No, I think it was 4.
I like knew how to do the first one, and then after that, I was like, I'm just not even sure. So, I'll just see what I can do.
Still alive.
Yeah, as long as that little green light is on. Do you see it right next to the camera?
Oh, yeah.
Then that means it's
I
recording.
didn't even see.
I just realized that right now.
Whenever I write phosphate on the computer, I always spell phosphate. Well, maybe we can just ask her. Maggie, I'm sure we can find it eventually, but
Yeah,
It's not popping up.
it'd be a good question to ask.
see, I mean, here in Wikipedia just refers to as it says a phosphate group attached to the 3 prime. But yeah, I don't know if that is just referring to in general. Yeah, we can ask her later on.
Okay,
It's not too important, I think. I mean, it's important, but I mean, it's not urgent.
right. Alright.
Something is really sticky and gross on this table.
Ew. Okay, so and then B, what are the 4 DNA nucleosides?
Haven't I read nucleotide and accident, so I just
Oh yeah.
Yeah.
So is it deoxyguanosine, deoxyadenosine, deoxythymidine, and deoxycytidine? Okay. And then C, suppose a genome of a newly discovered species of bacteria contains a double-strand DNA molecule composed of 24% thymine. What percentage of the genome is composed of guanine, cytosine, uracil, and adenine? And so it'd be 24% adenine because they match up, and then 26 of guanine and 26 of cytosine. And since it's DNA and not RNA, there'd be 0% uracil.
Is that what I thought?
And then D, suppose the genome of a newly discovered species of virus contains a nucleic acid molecule.
just think if we go on there. So then, just to clarify and see if we had an RNA molecule that was matching up with this exact same DNA molecule, then it should be 24% uracil, right?
Yeah,
If
so
it
it would
was
be
RNA.
instead of
Does
Yeah.
not
thymine.
mean-
Okay.
And then, so then it's just saying it has 38% adenine, 20% guanine, 35% cytosine, and 7% uracil. What can you conclude concerning the genetic material of this virus? And I put it's single-stranded.
I put this in.
Yeah.
Yep.
Alright.
Mr. Rooney.
Do you want to do 3 because you know what's going
Sure.
on?
Well, I had to ask a billion questions, but this question concerns the structure of chromosomes. The mouse version of chromosome is chromosome 1 is 197 million base pairs in length. How long is the DNA portion of chromosome 1 in meters? So you said you got this
So
one
I
right.
think so. You used the point. I did 197 million times 0.34,
Mhm.
and then I just moved it over to meters, and it was like 0.067.
Yeah, so yeah, that's what I did. What she told me was just to treat them as stoichiometry problems from chemistry. And you just
Okay.
have to find the conversion factors, which was
Yeah.
what I was having a hard time with. So is that what you got for party?
Yeah. Well, it's part of you said 0.6698 or 0.67.
0.067 meters.
Yeah, that's what it should be. I actually wrote it down.
Oh, you probably just missed it.
The
That's
0. Yeah.
Yeah.
Oops. Yeah. 0.06.
Okay. And part B. Oh, my answers are wrong on there though, but I know how to
Okay.
fix them. I know how to get the right answer.
Okay.
Based on what we learned in class, how many nucleosomes are found on the mouse version of chromosome 1? Assume that the average length of the linker DNA is 50 BPs, base pairs. So for that one, the conversion factor is that there's 146 base pairs per every nucleosome.
Okay.
And then you have to add in 50 for that linker.
So you do like 196?
196 base pairs for every nucleosome.
Okay, so then do you take your, what would it be, 197
Yeah, so
billion divided by,
so it'd be 197 billion base pairs. So you would put, I guess I have it wrong right here. So 197 base pairs and then you convert that to nucleosomes, so 196 base pairs for every nucleosome. Is that what it's one nucleosome?
Then what's the answer on that one?
It's
Okay.
about a million. So the exact answer is 1.00 to 1.01 times 10 to the sixth. I just said approximately 1 times 10 to the sixth.
Yeah, so is that what you did? You divided
So
the million
because
by
there's like
196?
about 197, one for then it's a million. So then it'd just be like 1 million,
Yeah. So
basically.
Right.
So for it's the range because it's 146 to 147, and yeah, divided 1.97 times 10 to the eighth over 1.97 times 10 to the second. That's how
Oh, times 10 to the second, yeah.
10
Yeah.
to the second, yeah.
Yeah.
And then from there, that's how I got that. And then
Yeah. Yeah, what helped me, what she told me is just treat them as stoichiometry problems, so just find the conversion factors.
Yeah.
So we go
Okay.
from base pair and then base pair to nucleosome.
Okay.
in that first one I was looking at that, I think I actually just did a calculation on that, when I do my calculations, I remember I put 3.4 and not 0.34 nanometers. But I guess I don't know because I was doing 10 base pairs. But I'm trying to see
So it should be 0.34 nanometers
for one base pair,
for
right?
one base pair.
So then for
And then 1 nanometer is 1 times 10 to the negative 9 meters.
okay. I think that's why I did. I did times a rotation, and yeah. Okay, that's what I did.
Okay.
Sorry. moving
Okay.
No,
on.
you're fine. Okay, the haploid human genome is approximately 3 billion base pairs in length. How many histones are predicted to be found within the haploid human genome?
So do you do 3 billion base pairs, and then you do 1 nucleosome
Right.
per 196 base pairs,
Mm-hmm.
and then
And there's 8.
there's 8 histones in a nucleosome.
Mm-hmm.
So you do like 1 nucleosome on the bottom
For
and 8
histones.
histones.
And that should give you the right answer, which is not that.
Yeah.
I did them all wrong, and then I saw that key, and then I was confused when I asked her, and she explained it. to me.
So in this case, the 50 base pairs doesn't really matter.
No, it does because there's 146.
Oh yeah, because you add 100. You
Yeah.
add the 50 to the. Okay. I got
You
it.
guys got 1.22 times 10 to the eighth.
We both did it wrong originally, so.
Okay.
Yeah.
But is that what's on the key?
Yeah.
Yeah.
And then the key has, I think, the
Yeah,
The conversion
multiplying,
factors.
Yeah.
it does.
So what I didn't understand from this was the linker. How do you assume that there's just one linker? Is it?
Did you
Because
see the
because-
picture where it's like this is H1, and then it's like 1, 2, 3, 4, 1, 2, 3, 4.
Is it
like
H?
that.
Is it H1 or is it? It's because there's 8 histones to every.
I think it is.
Whatever you call it.
I think
For
because
every
there's
nucleosome?
H2A and H2B, right?
Yeah, H2A, H2B, and H1, right? And H.
3.
H2.
I'm
Because
pretty
there's
sure.
2 of
I'm
each.
pretty sure that H1 is the linker, and then there's H2A, H2B.
But so for all of these, there's always one linker for every like haploid.
nucleosome.
So the picture, I mean, it might be out.
What picture are you talking about?
So there's this one right
Well,
here.
there's one linker per nucleosome,
Right. One.
and then
Okay.
there would be, however many
Yeah.
Oh.
in the 3 billion.
Yeah, so this is the linker region right here. So you got the nucleosome and nucleosome, and the linker region in between, and then between the next one to the next one.
Right. So what I'm saying is, so like what was the answer for this one for C?
For C, it was 1.22 times 10 to the 8th histones.
So you have histones.
histones.
Okay. So for part B, when you're calculating nucleosomes, how come that's the number of nucleosomes if you're supposed to have 50 base pairs in between each one of those?
Yeah, so each nucleosome has 146, 147, right?
Mm-hmm.
And then there's 50 base pairs on top of that.
On each one. So you're taking it into account when you end up dividing by.
Yeah, so each one, there's one linker region to each nucleosome.
I guess for some reason in my mind, when you just added it once, it meant you were just taking into account one of them. But okay, that makes sense.
No, it's just saying one nucleosome, you have one linker, and then 147 other base pairs.
That makes sense. Okay, for every one
46,
nucleosome.
yeah.
Okay.
Okay, and then
How many copies of the histone H3 are predicted to be found in the nucleus of a typical human somatic cell? I'm not even sure if that was right. I put 2.
I put 2 as well.
No, it's
because I didn't
6.1
know.
times 10 to the 7th.
How did you get that one?
So you do the answer that you got for this one, or I did a shortcut, I guess. I don't know if this is the way that shows it on
But
here.
I thought it said, okay, what I understood was there were 2 copies of each one for every.
But then that's not every
But
cell.
that's for every nucleosome,
Yeah, so
right?
yeah,
So how many nucleosomes are in
in
a
a
cell?
cell. Yeah, so how I did it was there are let's see. So roughly the haploid genome has approximately 3 billion. So that's one cell. So this is a haploid cell up here, right? So the somatic cell is going to have double the amount of that. And so I just took that and I times it by 2. But then it's only asking for the one type of histone. And there is two of that type of histone out of 8. So that's 1/4. So I just divided it by 4.
Oh.
So I just give
So you times this number that you got at the end for histones by
C.
2,
by 2.
and then
because
times it by 1/4.
and then it times by 1/4, yeah. Because
Okay.
there's 1/4 is not a histones because you're looking just the one type of histone, and then
Makes
somatic cell, not a haploid cell, so that's why it's 2.
So part C gives you the amount of nucleosomes.
But that's not how it shows it on here. What?
Oh, how does it show it?
So the actual, so that you can see like the whole, I just,
Can you put it this way a little more?
Yeah.
This
so,
way.
so this is what it shows here. So there's 6 billion base pairs
So
for a
it
somatic
just
cell.
doubles that
double the 3 billion,
instead
yeah.
of,
So it's basically doing the exact same math as up here, just with 6 billion instead.
and then the 2 instead of the 8.
Yeah.
Okay.
Wait, so where did the 6 billion come out?
So there's 3 billion base pairs, and this is a haploid cell. This is a somatic cell.
Oh,
So there's
does every somatic cell have 6 billion, or is that just for this
For
one?
this one. For this
Okay.
one, yeah.
And then it used the same nucleosome, and then 2. Oh, okay, that makes sense. You just
Okay.
did a shortcut.
Yeah.
Okay.
That makes sense.
Good things of getting a new computer in like 6 months. So,
Okay. Oh, see, those are actually.
okay, so 4.
Which molecules above are nitrogenous bases?
So I said guanine, uracil, and thymine for nitrogen
So
species.
we-
And B, I got adenosine triphosphate, uridine, and guanosine, for which contain ribose.
Where did
You
you guys
got
find that?
uridine, guanosine, adenosine, and adenosine triphosphate. Do I have an extra
Wait,
one
see
in
that
there?
again?
Uridine,
Okay.
guanosine,
Okay.
adenosine,
Uh-huh.
and an adenosine triphosphate.
No. That's right.
Where did you guys find that? Because I can't find it.
So it's just
The ribose is the sugar, right?
Yeah, so if nucleotides and nucleosides both have a sugar, right?
Mm-hmm.
Yeah.
So I just looked at the list in the instruction notes. And I just looked at those 2, and then I looked at the list and saw,
So you put every nucleotide and every nucleoside
yeah, from this
from
list.
the list.
Yeah,
Okay.
yeah.
Oh,
They go with the ribose.
yeah.
That's why it's uridine. Not, are we good for B then?
Yeah.
Okay. And C, I got adenosine, deoxythymidine, uridine, and guanosine.
That's
Yeah, so that's a,
what he got.
Yeah.
and then D, I got deoxycytidine,
Monophosphate.
yeah,
Yeah.
monophosphate. Sorry, I just didn't finish the word.
I didn't really want to write the whole thing.
Adenosine triphosphate and deoxyuridine diphosphate.
Okay, and then 5, I guess we're supposed to do it together.
Okay.
So, suppose a DNA strand has the following sequence. I'm not going to read that.
Alright, CTG. Okay.
Describe what it meant by the 5' on the left-hand side of the molecule. So pretty much, we kind of talked about it before. Like the 5' end is the one with like the phosphate attached to it. It's the 5' carbon.
Mm-hmm.
It's the -1, and it's the side. I mean, if we're talking about like replication, it goes to the 5' end.
Yeah, and on that end of that molecule, all the molecules on that side, it's the 5' carbons on that side.
Mhm.
Okay. Wait, apparently I write it down, do I? Because we're going
Yeah.
to need to talk about it. Okay.
And then
Describe what it is meant by the 3' on the right hand side. That's the same thing, but it's opposite. That's
The hydroxyl group.
hydroxyl group, and is that the negative or the positive?
I think that's positive.
Yes, positive. Yeah.
What do you mean by negative and positive?
So like in the phosphodiester bonds, it's the positive part of it.
Oh, okay. Yeah.
See, what is the sequence of a complementary DNA strand that would have the capacity to form hydrogen with the sequence above?
So just be the opposite. It would be TACGTGGC.
Oh, you have it backwards.
So I took 377, and we were always taught to write it from 5' to 3'
Oh.
direction, no matter what. And so that's just what I did. But I labeled it, so.
Yeah, that makes sense.
From 5' to 3'.
Yeah, that's
Yeah.
What, she has the same thing with
Because, like, we had to, like, send
Isn't
in
5'
our.
to 3' right here.
Yeah, but so you just do, like, it's right. It's just backwards. Like, TAC goes with the 5.
Mm-hmm.
ATG, TAC.
Wait, but isn't
So this is why I do it. Like you have to send in like primers and things when you like actually sequence DNA, and so you always write it in the 5' to the 3' direction, and so that's why I just went like.
But isn't that like counterintuitive? Like I mean, because you wrote it. Wait. Where's your answer?
Yeah, so it matches up because it goes like that.
TAC. It just goes like this.
Mm-hmm.
Life's got me. So.
I have the same answers as he does. Yeah, that makes sense. Look at you going next, Romano.
I just
No.
like.
Not yet.
It's
We'll be
always
done
just,
probably
like,
in
at
like
the
10 minutes, maybe. Give or take. I'm in the Benson. I'm in that room that we always study in the first floor. Love you. Bye.
Alright.
Wait, because I'm still confused though with how.
So what she did is.
Yeah, I kind of see what you did, but it looks like because
I have the same answer as you.
I just wrote it
Yeah.
in that direction. I wrote it complementary, so the 3' end matches with the 5'
Oh, that's right,
end.
because it's switched around.
Right.
Oh, okay.
Yeah, so it's 5' and then 3'
That's
on
right.
the other side.
I'm just like, that's just what we were taught.
Yeah, because it's gonna be 3', 5'. Okay. And then you wrote 5' to 3'.
Mm-hmm. Yeah.
On there. Okay.
So mine would be the same if I like read it off. It's gonna be the same
Yeah.
as what you guys said. TACG.
Understood.
Yeah.
You're sophisticated.
You just get used to things. I feel like as like different classes you take, and they're like, do it like this. You're like, okay, well, I'm gonna do it like that.
Yeah. Have you guys seen 321 yet? Bio 321. I hear you have to go to like do like a camp thing over.
Oh, that's what they were
That's what
talking
they were
about.
talking about. Yeah, because I heard someone else talking about it and they said they hated it. I mean, it depends on your teacher probably, and then also, I feel like
That's
don't
just like
learn
a really
anything.
weird thing.
I think it's super weird.
Like, can you require kids to come do something on a day where it's not classes?
I don't know.
I'm not sure.
So like if I were to
Because
do
I
that
feel like
this
you
semester,
couldn't
I'd have
make
work
a
and stuff.
test just on Saturday. You'd have to have it open on like Friday or Monday or something. So it's
Yeah.
kind of weird, but I don't know. It'd be cool if they did for like extra credit, but not for like
An actual assignment.
an actual assignment. Yeah,
I think so.
Yeah, that's weird.
that's really weird. Okay.
Okay.
D. Suppose a molecule from part C is able to form hydrogen bonds with the sequence above. How many turns of the DNA double helix would be found in this molecule? Wasn't it 10 base pairs per turn?
Yeah.
And so there was like 25, and so it'd be like
2.5.
2.5.
Yeah.
Yep.
I just put 2 because I was thinking like full turns, but that makes sense. It'd be 2.5.
How long has it been? But it's been. What happened? Is it still recording?
Should be.
Green
Not
light's
mine.
still on.
Yeah, it's still going. 26 minutes.
4 more minutes.
We can make a rap about the DNA sequence. Okay.
What are like the extra things that she said? Because I know she added things to like problem 1 instead of just saying a phosphate group. She said like a phosphate group.
Yeah, there was like a
something,
whole
something,
paragraph.
something. Yeah.
Let me see here. Perspective 1. No. Número 1. Okay. So that was for which one?
What are the 3 chemical components of a nucleotide?
1 to 3 phosphate groups. Pentose sugar, either ribose or deoxyribose in a nitrogenous base. DNA contains blah.
Oh, and it just had like the bases listed. Okay.
And then were you asking about like the carbons, the 3' or 5'?
Yeah, and then on number 5, what did she have for like all the things she said?
She said basically what we talked about. Said the 5' carbon of both ribose and deoxyribose is attached to a phosphate group. Phosphate group on the 5' carbon of a nucleotide functions during DNA replication and transcription. Covalent bond is formed between the 5' phosphate group of the incoming nucleotide and the 3' hydroxyl group of the existing nucleic acid strand.
Okay, and hydroxyl is positive because it's attached to 3.
Yeah.
Okay.
I think it wouldn't be like a bond that wouldn't be as stable as it is. I wonder why it is.
Yeah. Especially like it's a pentose, which is fairly stable, but it's not as like.
But I guess like
stable
the
as-
phosphoester bond making the positive charged oxygen, because I feel like it wouldn't be very stable. I feel like it one is separate.
Mhm. I don't know.
I don't know.
Maybe it's just like a good
Is that my
enough
phone playing?
balance.
Or is that you? Oh, that
Mm.
was my phone. I kept hearing a voice. I was like, I didn't think my
Who's in
phone's been
here?
playing music this whole time.
That's funny. Are you guys ready for the test?
No.
I'm gonna be studying. I was gonna study more today, but then I went to that. Did you guys go to that forum thing they had
Mm-mm.
with President Irving? It was really good. It was so packed. I literally sat on the floor and
What did
yelled.
he talk about?
Because it was just in one of the rooms in here, wasn't it?
It was the really big room though. It was 108 and 110. So none, it was a big room, but I mean, like it was just packed. Like people, like I was on the floor, there's other people on the floor too. We talked about like a lot. Let's see. We talked about he doesn't really have a healthcare background. We just talked about his story of he was initially was denied because he went to Harvard Law School, but first time he applied, he didn't get in. It's also the wrong notebook. But
This is President Irving's son, right?
yeah, he is. So he talked a lot about how God has a plan for your life, and you can discover that plan, and then that was kind of how we started off, I guess. Was talking more about that and trying to, or striving to follow God's will, I guess, or not follow God's will. I mean, yeah, but that wasn't really. I'm trying to remember, but then he, I don't think that was part of the planned presentation because afterwards I wasn't going to say that, but this one was spiritual. It was really good though. And then he talked about the economic side of things and also the balancing family life and profession and choosing a profession that is economically like you can, if you do have to go into debt, you can get out of that debt and like not unnecessarily going into debt and like along those kind of lines, economic and family life with career. Yeah, no, it was really good.
That's awesome.
So he went to law school.
Yeah, so he majored. It was funny. He was like, "Don't." He said like a million times. He's like, "Don't do what I did." He just kind of like had this idea that his dad went to Harvard Law School as well, I think, and so he majored in geology at Brigham University. And then there's only about 3% of people that apply to Harvard Law School at first that haven't. So only 3% of people that are accepted in the Harvard Law School are like right out of college. Most of them have done like got an MBA or done something else, work or something like that. And so, so he ended up majoring in geology and then got his MBA. Hey. And then he went to Harvard Law School and stuff. I don't know if I answered your question or not because I kind of forgot what it was.
I don't remember.
But I think it's been 30 minutes. So.
Yeah.
Probably.
Did it log out again?
I don't think so. Maybe? Oh, no? I'll press stop.
32. Woo.
Stop?
Yeah.